to the mass spectrometer. So we are going to talk to you about proteomics based workflows and maybe refresher of some of the technology. For example, how you can prepare the samples, in which way you can now separate the complex proteome mixture on the gels based on the one dimension or the two dimension properties, how you can identify the important proteins of interest, or can you do shotgun proteomics, directly prepare the sample and run them on the mass spectrometers to look at all possible proteins. Once you've identified these proteins, or even sometime you might be interested to look at the metabolites or metabolomics, and eventually you want to validate your targets. So that's where you'd like to look for targeted proteomics approaches. So we're going to walk you through on many of these interesting and exciting technologies of the field of proteomics. But before we start that, let me introduce you to our TAs who have been working very hard with you over the last few weeks of the course. So let me share my screen, and one by one they will introduce themselves first, so that you are aware of to whom you are talking, and then we will start with the lab activities. So today's live session title we have made on the gel based and mass spec based proteomics, and let's start first with Amrita Mukherjee. She's a senior PhD student, and Amrita, you can talk about your work.
Good afternoon, everybody. I am Amrita Mukherjee, and I am a final year PhD student working with Professor Sanjeeva Srivastava. So I have completed my bachelor's and master's from University of Calcutta, and currently I am working on cervical cancer tumorigenesis, finding the protein biomarkers in disease progression, and dealing with the cancer resistance in Indian population. So in this particular course, I will be a TA of MOOC Bioengineering course, and along with Mr. Ankit Haldar, and looking forward to meet with you all.
Great. So Amrita has already been well experienced in teaching to IIT Bombay undergrads the bioengineering course, and she has also done the MOOC course. So again, some of you are already interacting with her, but others are also encouraged to take the opportunities to talk to the TAs and the instructor, so that it just does not become like a recorded video based lecture. Rather, you have opportunity here to interact with the TA and the instructor, and you can make use of those platforms. So great, welcome Amrita, and let's move on to Ankit Haldar now.
Hello everyone, good afternoon. So I am Ankit. I am a fourth year PhD, STEM PhD dual degree scholar at IIT Bombay, and I am also a PM recipient of PMRF fellow, and so my current research focuses on one of the kind of brain tumor which is meningioma, and I will be the TA of the bioengineering courses, as well as I am the PMRF TA who conducts the live session for the Interactomics. So yeah, so it's always nice interacting with you all, and looking forward to interact with you more. Yeah, thank you.
So again, Ankit has been already interacting with you every week live on the journalistic matter of work. But he has also taught multiple times at IIT Bombay to the bioengineering students. And this can be a good opportunity for you guys to interact with him. Also learn a lot of new ways of data analysis, which he is quite specialized in. How to take this type of proteomics and multi-omics data and do much bigger big data analysis and make sense of the data for visualization. So, all right, welcome Ankit. Let's move on to the last TA, Ayushi.
Good afternoon, everyone. I am Ayushi. I am also a final year PhD student under Professor Sanjeeva Srivastava. My PhD project involved metabolomic and proteogenomic characterization of high-grade gliomas, and I'll be your TA for Interactomics course, and hope you are enjoying the course, and we'll be interacting more during the course. Thank you.
Right, so yes, Ayushi is the TA for the Interactomics course, and she has been working on multiple technologies, from proteomics to the metabolomics and proteogenomics, and again looking at various biological network and interaction analysis. It will be really interesting if the students are making best use of these resources and the TA available, and both the courses can get benefited from these interactions. So with this brief introduction of the team, and of course, you probably know me, you have already seen my videos, but I'm the instructor of your course and will be with you throughout the course journey. We are quite accessible, even we have my Telegram group. If you are, have not joined, please do join that so that you can be benefited from it. The way we conduct more courses, we don't feel that you are just, somewhere online. We always feel like, you're part of our academic system, and we place a lot of emphasis and good reverence to all the students who take the course. In fact, many students who do these kind of MOOC courses, we give them opportunity to come to IIT Bombay for some internship program, some workshops, training program which we conduct. Just now we conducted one big international conference, Advances in Proteomics Technology APT 2024, and we had also some workshops there. We gave opportunity to the MOOC toppers of my previous courses to come here, almost free of cost, to come and attend this workshop and training program. So again, if you are really keen to learn this field, we'll always encourage you and provide you some right resources and platform so that you can build further from that. And my own level also benefited from such interaction in the past, when many of the MOOC topper students have actually, formally worked in my lab and eventually did a PhD and did great work, with this kind of interaction. So that's the, idea for learning. So don't feel that you're only watching the videos and we are only virtual available. We are definitely very close, and we would like to make it more stimulating journey together. So let's move on to the next, activity, which is essentially the sample preparation. So Amrita is going to start giving me some basics of the sample preparation, which slightly changes based on the context of the sample, like what kind of sample one is working on. But overarching scheme, the overall goal remains same, that how best you can get rid of the, other than protein, any nucleic acid, lipids, carbohydrates, and most detrimental the salt, and how best you can break open the cell, maximally how much protein can be out of that, and then in which way you can further digest the proteins to make peptides, clean them up, and run in the instruments, right? So the sample prep remains the key area for the proteomics, and I'll hand over to Amrita now to walk you through the steps and showing the lab protocol directly. Amrita, please go ahead.
So for the sample preparation part, Amrita, now you can take over and start presenting.
Hope I'm audible. So today I'm going to walk you through the basic sample preparation steps which happens in our lab. So our samples are basically stored in a -80 freezer, be it tissue sample, be it serum, be it a cell pellet. So this is a -80 freezer. Currently the temperature shows us like -86.5, and samples are stored over here. I'm going to take the samples out and going to show you how we can the from every sample. So this is one of the cell line sample we have. So this is the pellet extracted from a cervical cancer cell line sample. Now to extract proteins out of it, we need a particular lysis buffer. So here we are going to use 8 molar urea lysis buffer. Into it, we are going to pour approximately 100 microliter of urea lysis buffer into it and mix it well. As you can see, so that the pellet is completely dissolved in the lysis buffer. Mixing well is very important at this particular step, because you do not want to leave any pellet residues behind. Now, once you mix it, you have to make sure that it is properly mixed. The next step is the sonication of the samples. So here is our sonicator. So we have applied the settings that we are going to sonicate for 5 seconds of pulse in a 55% amplitude throughout the 1 minute. So, but one important point is that when you have poured the lysis buffer and when you do the sonication makes a lot of heat. So that could be detrimental to the sample you have. So you have to place the tube where you have the sample on it and tube on ice and do the sonication. So here, if we start the sonication, if you can see that right now
that the cells are getting sonicated. So the purpose of using the sonicator is that the efficient lysis of the cells, so that the protein, the intracellular contents of the cells comes out and we get as much as protein content as we want. Now this will go for next 5 minutes, but once the sonication is completely done, so we assume that the sonication has happened for the next 5 minutes of cycling, you will get a clear, crystal clear solution like that. You have to perform the sonication in multiple rounds unless you find a crystal clear solution like that. Once you find a crystal clear solution after sonication, we will do a centrifugation step. So this is a centrifugation step where we are going to centrifuge the sample in 8000 RPM for 15 minutes, and it is very important to maintain the temperature at 4 degrees centigrade. Otherwise, it will be detrimental for the proteins that you have just extracted. So we have already sonicated, we have already centrifuged previous samples, and it looks like this, and we are going to collect the supernatant from it. Now, once we collect the supernatant from it, your lysis is now prepared, and now you can store it in the -80 freezer. From that, I have take the sample out. Now, once you have made the sample, the next step will be the quantification of the sample. Now, we have on a daily basis, we apply the sample by Bradford assay. So what we do is that the Bradford assay, we use the Kumasi brilliant blue dye, and the Kumasi brilliant blue dye, when it is mixed with the protein, it will give you a blue color, and it is the intensity of the blue color is directly proportional to the amount of protein present in your sample. So, for example, this is your protein lysis, and this is the Bradford reagent we have. We pour 1 ml of Bradford into the unknown sample that we just extracted, and the protein concentration is not known. So you pour 1 ml of Bradford, you vortex it nicely and incubate at dark for 10 minutes. Now, once you incubate the sample at dark, you have to measure the absorbance value of the sample, and we are going to show you how we are going to do it. Now, to quantify an unknown sample by Bradford, it is very important to prepare a standard curve. So, for that, we take multiple concentrations of BSA, starting from 2 μg to 10 μg of BSA, and prepare a standard curve using the Bradford assay. And using that standard curve, we quantify the unknown protein concentration of the unknown sample. Now, as you can see, we are going to use this particular multi-scan to do the protein quantification. We have a ScanIt software installed in our system to run the multi-scan software. So, as you can see, we are going to place our plate that has the multiple different variants of these as well as our unknown sample into it, and we are going to take the absorbance at 560 nanometer. Now, once you have taken the absorbance, you will get the output value as like this. So we are going to put, so these are the absorption values from different concentrations of BSA that we got, and we have done the experiments in replicates. So we have got replicates, you have taken the average value, and we plot the average value of absorption average value, and you get the standard curve. So a standard curve, you will get something like that, where you must have a R square value greater than 0.9, And from that particular equation, you will be able to estimate the concentration of an unknown protein sample. Now, once you have done you have extracted the protein, you have estimated the concentration of the protein, then the next step will be to check the quality of the protein in a SDS page. Now, my colleague Ayushi, she is going to take it forward while explaining you how to do the QC in SDS page.
All right, thank you, Amrita. So before we, go to the next step, let me just give a short overview of the gel-based proteomics. And the idea is whatever you want to do in proteomics at different stages, you have to be familiar with the gels, right? And I'm sure many students who are watching us live and who are taking the course have already understood the importance of various type of gel-based proteomic platform. So when you're starting from the samples, the very first thing is you want to look at qualitative profile of the sample, how good the protein extract what you have obtained looks like. Here in this case, we are talking about a complex mixture, right? We're talking about proteome. But let's assume you are only doing a protein purification for one single protein. So that time you are just going to run on a gel, LGS page, and look for that one prominent protein band of your interest. In case of the complex proteome mixture, you want to look at many distinct bands throughout from the high molecular weight to the lower molecular weight. And you want to be sure that they're not degraded. They are sharp bands and they are representing all the range of molecular weight. So that's something which can be utilized as a benchmarking for your qualitative good sample preparation. Sometime your sample, I started talking about bacterial sample in this case, which may not have too much bias, but sometimes if you're looking at serum protein or plasma protein, you will look for a lot of abundant proteins will be like big block over there, and then remaining many things will be quite affected because of that, so very low abundant. If you're looking at some sort of plant sample like plant leaf, ribisco has very humongous band. So again, the gel can give you the good profile first hand to know that the proteins are separated uniformly, or are there some abundant protein or interferences there which is affecting the quality. And once you do that, and if you want to do now proteomics and identify the samples for the protein identification, you can even simply you can now take the entire lane of the SGS page and you can chop down that to the maybe 10 to 15 pieces, right? That entire gel. And each of the gel per part, you can proceed for the in-gel digestion. And now, in a way, you are actually fractionating your complex proteome also by running on the gel. And then you can now utilize that to run them maybe 10 samples from the one sample now, 10 fractions you are going to run on the mass spec and combine the data that will go give you much larger proteome coverage. But then sometime your goal is to also further comprehensively separate the protein on the gel using different principal type, like not only based on molecular weight, but also based on their isoelectric point. So that's where the technique which is shown on the screen is two-dimensional gel electrophoresis developed by Angelica Gorge, who developed the immobilized pH gradient strip actually, and the technology was developed by Close and Farrell in 1975 independently. And this technique gives you, as you can see different images here, lot of ability to separate the proteins based on the molecular weight and pI. So a complex mixture can be at least well separated on the gel. If you're looking at, let's say, a complex mixture of the pull down assay, you're looking at a complex which is biologically very interesting to you. And now you want to further study that. If you separate them on the 2D gel, you can get all their possible, different protein as a mixture to well separated, and now you can excise those spores. Or in this case, which I'm talking to you about the bacterial proteomics, when you're looking at the drug treatment, one is the untreated, one is the treated. You run them on the separate gels. But again, here the major challenge will be if you're looking at the entire proteome, the coverage might be limited. And the detection of the signal with the Kumasi and these things will also be challenging. So therefore, sometimes people use the technology known as the DIGE, difference in gel electrophoresis, where you can label your control and treated sample with Psi 3 and Psi 5, And you can also make internal pool, internal standard with the Psi2 dye and mix them all on the same tube after labeling. And then what you see here, after labeling, you are separating them on the one gel and scanning them on 3 different wavelengths to identify 3 different types of the images, which can be utilized for the further data analysis. This is one way of doing the quantitative proteomics, gel-based proteomics. But in the context, I have been also giving you equal hint that even if you want to do directly mass spec, gels could be a good intermediary point for you to do multiple things. One, you can do qualitative analysis for your sample prep. Second, you can use them as a fractionation way to cut down the bands and look at entire of the gel to separate the complex proteome. Third, you can also utilize even what you see here, something like isoelectric focusing unit. If you want to utilize them for fractionation of the proteome based on the isoelectric point, you can use the off-gel fractionator, a similar principle, when you can take the IPG strip, and put them in the IF field, and you place your protein in the solution form, and on top of that you can place the plastic cup, and then the proteins will move, and then you will get the protein separation based on the PI value in the respective cups. So then now you can take pH 4 to 4.5 in one of the fraction and 4.5 to 5 in next fraction. So now you got again fractionation done with the PI value. This is another way of doing it. So three tips I gave you. Fourth is, if you're looking at the complex mixtures, you can separate them on the 2D gel, and that can give you much better resolution. So even if you are not interested to do the complex proteomics on the gels, still there are many innovative ways where gel can be utilized to improve the quality of your entire proteomics workflow. So, having said that, let's now move on to the lab, where Aishi is going to show you the something as basic as the GS page, and then we'll talk about the 2D gel and other technologies. All right, Aishi, to you now.
Thank you. So we have already a crafted gel here. Sanjay, if you can focus on you. So we have a gel, MGS gel, and now I'll be showing how you can set up the gel for the electrophoresis. So we have very smoothly take out this gel. As you can see, there are 10 wells. So we can load at least 10 samples here. I'm going to put this glass plate inside this cassette. So this cassette can hold at least 2 gels. Right now we have only 1 gel, so we are going to put 1 gel at one side, and on the other side I'm going to put the dummy plate, which is just the replica of the actual glass plate. So you have to make sure that we are closing these clamps firmly, otherwise the bubble will leak out. So that's how we have prepared a setup, and this will go inside this tank, which is already filled with the running copper. So like this I have placed, and before loading the sample, we'll very nicely remove these combs so that all the wells, you can load the samples in the well. And then we have some sample here, which I'm going to show you how to do it. So you can take 10 or 15 microliters of the sample, which is with your loading die, and then while looking at the wells, you have to load the sample. Very slowly you need to load the sample. Here I am loading the loading die. And once the samples are loaded, you can just put the electrode over the unit. And then, yeah, you can run the scale on 90 volts. On constant voltage, and then we'll run it. So for the SDS page, the proteins are with the SDS, which is the negatively charged. So it will be uniformly, proteins will be negatively charged, and then protein will be separated based on the molecular weight. Thanks.
Well, all right. So since I've already talked about the SDS page, I can take it from here, and further separation of the complex proteome could be done on the based on the 2D electrophoretic which are talking. The same setup here. So same setup can be used, but we have to first utilize here the isotopic focusing IEF. So I think what setup is kept here is for the rehydration of the isotopic focusing, which will do with the IPG strip, and this particular tray is basically a rehydration tray. So one first it starts with your IPG strip, which is different type of length which we have. Sometimes it is pH 3 to 10. If you are not sure about the physiological context, then you can start with a broad range. Or sometimes if you are very much narrowly looking at the actual biological physiological range, then 4 to 7 is actually better to start with. It's always important that you know how well you can separate your complex mixture. So that's where having the larger unit, larger IPG strip can be more useful, and that's where these kind of trays gives you ability to look into separation based on the up to 24 centimeter long strip. But the strip could be of various lengths, right? So what we see here now, I think this very large IPG strips where you can separate the complex mixture on the very much with the higher loading of the proteome, right? So the first step goes into rehydration of the your IPG strip with the protein. Depending upon, you can use either active dehydration or passive dehydration. The major challenge here would be how well you can separate your protein on IPG strip using the isotopic focusing. And this is where this unit comes, the IEF unit. And while there are many instruments available, but this particular instrument gives you the close covering, so that if you are also labeling your proteome with the fluorescent dyes, it will avoid any kind of quenching and the leaching out. So therefore, this is I think good thing if you are planning for a dye style experiment with the fluorescent labeling. So workflow for gel based remains straightforward. You are starting with the protein extraction, which Amrita explained to you. You go again with the IEF, isotopic focusing, with the this setup which we have showed you now. Once this is done, then you can go for the further separation based on the SDS page second dimension. But again, if you are looking at the complex mixture to be separated, that this kind of unit, which is known as eta and DAR 6, can be utilized well, because now here you can, if you show from the dollars, you can really separate the complex mixtures with the 6 gel, and they can be even up to 24 centimeter long strip which can be utilized. So this workflow is quite standard, but at the same time has limitation in terms of what the maximum coverage you can obtain from the proteome. If the sample is small, like bacterial some proteome you're looking at, this could still be a good place to go. If you're looking at complex human sample or other kind of complex culture sample, then you might want to explore more of the direct mass spec based approaches. But again, as I was mentioning to you, it's good to be unbiased and utilize the best capabilities of different instrument platforms. So for instance, you can use IEF for the separation of the complex proteome and fractionate those and use them to run on the mass spec. Likewise, you can use the SDS page and chop down the gel pieces and cut those to separate the fraction, and now you can run them on the mass spec separately to generate more coverage. So these are the different ways when you are looking at how to extract the protein, how to do the qualitative check, quantitative check, and resolve the complex mixture, right? But then next step will be, you got these bands or you got protein in solution form. How you are going to run them on the mass spec? Can you directly take them? Probably not, because now we are still talking about the protein. And to run them on the mass spec, you need to digest them and make them peptide. So next exercise which we are going to talk to you about protein digestion. both in gel and in solution based activity, which I will explain to you briefly. What do you want?
So, as Professor Srivastava said, that we cannot inject the complex mixtures of protein to the instrument, mass spec instrument. And before that, we need to digest the protein, complex protein mixture. So, for example, if we have this particular complex protein mixture in this sample, to digest the protein, we have to go through multiple steps before that. So we are going to perform the digestion using a very common digesting enzyme called trypsin. But before doing that, we have to prep the protein samples for the digestion. So first step will be the reduction of the proteins. So proteins are very complex 3D structure. So there will be disulfide bond, and digestion will not be possible if the proteins are in a 3D structure in a compact way. So we have to make it in a linear structure so that every single site is accessible to trypsin and the digestion can happen smoothly. So for that, for the reduction purpose, what we do is that we use T-cells. T-cells is a very nice for using for reduction of the sample. So what we do is that we take the complex protein mixture and we add the T-cell into it. T-cell is used as a reducing agent, and we incubate the T-cell in 37 degrees Celsius for 1 hour. So this incubation step is very important because this enables you a proper reduction of the complex system you have. Once the reduction is done, then you need to make sure that the protein does not fold back to its original state. So we'll alkylate the areas where the reduction has been performed. So for that, we'll use the iodostamide. So iodine is used to perform the alkylation. So these are the series of the steps to prepare the protein before your digestion. Now we add the IA and we will incubate it for next 15 minutes in room temperature, but at dark. Because the IA is light sensitive, that's why the thing has been done on dark. Now, once you are done with reduction and alkylation, you have to pH match of the reaction mixture. So, for that, we use a kind of buffer, which is the digestion buffer. We are going to add the digestion buffer into it till our pH comes at the optimum pH where the trypsin works, which is the pH 8. So, we'll keep adding the digestion buffer and we'll check the pH in a pH strip or something like that until it comes to the pH 8, and then we'll add trypsin onto it and incubate overnight at 37 degrees Celsius for the proper in solution digestion. For the in gel digestion, the step will be something a little bit different, where we have the SDS page in our hand and we'll cut the pieces of the gel. You can see this particular gel in your hand, and we need to perform an in gel digestion. You have to excise the particular band out which you are interested of, and then we have to perform the rest of the steps to do the digestion. So the next step will be explained by Professor Srivastava.
Yeah, so Amrita, I think it's a good idea to also briefly talk about the peptide quantification, not to show the process, but that's I think very important in the proteomics. So again, like if any sample you want to now inject in the mass spectrometer, you need to know what is the amount of protein available, right, and the peptide available to be injected. So the process I just explained you the in solution in gel digestion, but now you got the peptide and you need to do some cleanup step to remove the salt and other impurities. And then you get some, a clean peptide. But how much peptide yield you have, so that's something which you have to again do the peptide quantification. Given the short time, I think what we have, probably we'll skip that now. But the same way what she showed you the protein quantification, similar setup can be utilized for the peptide quantification, but with some changes of the way you can calculate. So now let's say that you got some good clean dry peptide of around 1 microgram which you want to inject in the mass spectrometer. So you have multiple options of using different type of mass spec technology. We'll just first walk you through with one of the very high-end instrument which can be utilized for the deep proteome analysis. It means you want to really separate the complex proteome mixture based on different type of principles involved in the mass spectrometer. So Ayush is going to now take you through arbitrary fusion and show you the setup and how to separate the proteins using mass spec, LC-MS/MS.
So we are in the mass spec room of our proteomics facility, and we have 3 mass spectrometers here. I'll be showing you the one which is the arbitrary fusion. It's a tribrid mass spec. Tanmay, you can show, and this mass spec is coupled with the nano LC, which is thermo easy nano LC. And the mass spec usually have 2 solvents, solvent A and solvent B. Solvent A is 0.1% Fa, which is usually for the binding of the peptides with the column. And then solvent B is 80% ACN, which is to elute the peptide. So now I'll quickly show you how we can place the samples here. So this is the auto sampler where many vials are kept. So we fill our peptides in these small vials, and then and this is the auto sampler, so it is robotic. So you have to just place these are the vials where we place the peptide, and it will go to here. And then we'll close the auto sampler. The samples are inside, and now I'll show you how we plan the runs. So to start the run, this is scientific Excalibur software where we can line up the samples. So as you can see, few details we need to fill here. So for example, you need to provide whether the sample that you are going to run is a blank or a QC sample or your unknown sample. So in the sample tab, you can mention the type of the sample, and then whatever file name you want to give, you can put the name. And the output file, which will be the .raw file. So you need to provide the path where this raw file will be stored, and then you also need to provide the method. So whatever method, let's say if you are going to perform PRM, then your PRM method will be here, and if you are going to perform DDA or DIE, accordingly the method you have to set here. And then in the auto sampler, if you have put your vial at the A1 position, you can put the position value here and how much volume you want to inject. So let's say for the blank we have mentioned 8 microliter. So the auto sampler will take 8 microliter automatically from the auto sampler. So likewise, if you fill the details and whatever sample you want to run, you have to just select it and press the and you can start the analysis. So this is how you can plan the runs. And now I'll show you one run which is already done. If you see here, this is the chromatogram of particular sample, which is the cisplatin sensitive cell line. So that's how you can see the one chromatogram will look. As you can see, this is 120-minute gradient. So on the x-axis, you can see the times in 120 minutes. And on the y-axis, we have relative abundances. So I'll show you how you can check the MS/MS also of this data. So you have to keep zooming in. And as you can see, here I'll place the cursor. And you can count the MS/MS, how many MS/MS is there in your one MS. So likewise, you'll get how much your data is good. As you can see here, we have so many MS/MS. So you can also check the quality of data by checking the MS/MS in your data. And you can also check the intensity of from here. Yeah, here you can see the intensity is in e to the power 9. So from so it's 2.34 e to the power 9. So intensity, if the intensity is in e to the power 9 to e to the power 10, it generally considers good. One more thing which I want to mention here is, generally we start with a blank. So whatever sample your sample is running, it should be followed by a blank run so that the previous residues of the sample can be eluted out, and then you can plan your actual sample. So we usually follow one blank before and after each of the samples. So that's what we follow. And we also follow BSA PRM as a QC sample. So whenever we start the run, we usually start with a QC sample followed by few blanks, and then we start the actual sample. So that's how you can plan the run, and you can also visualize how your chromatogram looks and what are the parameters that you should actually look at to consider a good run. Thank you, Tanvi.
Yeah, so again, like, what you see right now, that does not make any sense unless you analyze the data. And that's where good databases and the data analysis workflow becomes important. Only what you have seen just now can give you visual, qualitative, feel of that, okay, the run has completed properly and the chromatogram looks good. The spectra looks quite dense and intensity is good. That's indirect indication that, your data eventually should come out good. But then you have to take this raw data file and search again the databases. And then you look for, okay, what is the proteome coverage which you can obtain. And then how confident you are for that proteome coverage will only come if you know that different type of statistical ways of filtering that data out. So that can be for some other day. But right now, let's say now from the same sample, you want to further look at the metabolites and you want to analyze that. So another instrument platform which we have, which is a Q-executive mass spectrometer with HPLC, that can be utilized. And Ankit is going to walk you through that workflow.
Yeah, so now we are seeing for the proteomics one, proteomics how the different proteins can be analyzed to one is another instrument that is two examples of the that is an instrument which contains two mass analyzers, one is the quadruple and the molecular. So now this is coupled with HPLC for the LC part. So in the proteomics we have seen that it is coupled with nanometric with the flow rate in the nanometers. But here you can see that it is in the 1.350 minutes per minute, which is having a higher flow rate for the separation of the molecules. Now in the metabolic storage contains the solvent system again for measuring the fecal segment, and this is the sampler autosampler where one can keep the samples, and here this is the column compartment where the columns are there for the molecules to get separated based on the variant where the molecule gets bind to it and then get eluted out slowly, which is getting ionized. So now if I just give you a brief overview of the techniques, as you know that in the mass spectrometry one we are measuring the MYZ. So what is the major difference between the proteomics and the metabolite is that once we identify the MYZs of a particular metabolite, we can do the molecular weight, we can deduce the molecular formula of that particular metabolite and then can finally try to attach with it. For example, glucose as 180 and if it is charged as a +1, so it will be having 180 +1 or 181 and we can say it as a glucose. But for better understanding that whether that particular metabolite is the same or not, we do another fragmentation which is also done in the proteomics for deducing the amino acid sequence. So similarly, the MSMS spectra led us to understand that what actual metabolite is. So I'll just show you one particular chromatogram. So this is at the MS1 level analysis with a 20-minute gradient, where for a particular gradient which has given the separation of the particular metabolite from the sample. So you can see that here the MYZs have been identified where the mass is and the charge. For example, here you can see that the charge is 1 + 1 state and the 202.02. So now after going to understand all these particular at the MS1 level, when we do MS2 level and we can see the image fragmentation pattern, then for that particular fragment that which is one of was having for the 202, we'll have separate smaller fragments, and from the fragmentation pattern we can understand that which particular metabolite it is. So this is just the basic principle, and also I like to show you the particular gradient which we were talking about. So on the basis of the different solvency steps of different hydrophobicity, so we try to give the instrument that what percentage of particular solvent is mixed to achieve what percentage of medium, which will lead to the emission of the metabolites from the samples. So the major goal or of our thing is to have the proper dilution of the particular metabolites and certain over the certain period of time, so that it gets ionized properly, and it also gets detected after separation from mass analyzer and gets detected in the detector. And once it is detected on the basis of the MYZs and the MSMS
understand what is the metabolites, and also you can see that there is a intensity. So with that, we want to also try to quantify the metabolites present in the sample and try to see the differential expression of metabolites. Thank you.
All right, thank you, Ankit. And so what the student will appreciate that same technology platform of LC-MS/MS which we are talking can be utilized for the global proteomics or even global metabolomics as well. But now let's say you have identified hundreds of leads coming as the protein or metabolite, how to validate them? And therefore validation becomes quite challenging and important. There are many methods based on the antibody which you might be aware of, Western blot, ELISA, immunofluorescence studies. All of them are very robust, but they require good antibody availability. If you do not have antibody, because let's say you are trying first time a new proteomics initiative, and you have identified some 5 good targets for which there is no antibody available. So then you have no quick way of validating your leads, right, on the more number of samples. So a new methodology which is being used now, you can also try that, is the targeted proteomics, where now you know the peptide which you have come out of your discovery data set, and the peptide of those proteins which is of most interest to you as a biomarker. Now you can select those and run them on a triple quadrupole based mass spectrometer. So now last live demonstration will be on the triple quadrupole TSQ artist instrument, which Amrita is going to show you shortly.
With the HPLC system, and as you can see here, we have the sample. This is the auto sample where you can put the sample and then the system takes up the sample and the mass spec is being analyzed the sample. Now, what happens in targeted proteomics is that so as you already have then you instruct the instrument to search for specific peptides. So you instruct the instrument to search for specific peptides, and you look the only peptides that have that desired MYZ value. So here, as you can see, so here in the system, so this is an example of how the targeted analysis experiments have been performed. Once you have run the samples and you get the raw data, we use the Skylight software to analyze the raw data. This is one example on how you analyze the raw data. Now we are showing a particular example of COVID positive and COVID negative samples, and we are going to show the expression of a particular protein on the differential expression of a particular protein on this particular sample. So if the protein that you have already got is as a lead from the shotgun proteomics, now to make sure to do that, we first run a synthetic peptides throughout the samples to make sure that there is no instrumental bias. Instrumental variability is nullified. So we run the synthetic peptide, and as you can see, the intensity of the synthetic peptide is very much uniform in all the samples, in the COVID positive and COVID negative. And then when you come to your actual sample, actual desired peptides, where the intensity is supposed to be varied, you can see that in the COVID positive samples, COVID negative samples, the intensity of a particular protein, which is called the surfing jiva, is very less as compared to the COVID positive samples. Now, this is how the chromatogram of this particular peptide looks like, and these are the intensities. Now, with each intensity, you are going to give it so we are going to compare our data with the ProSec library, and the library gives you a .p value, which tells you the significance of the occurrence of this particular peptide in the sample. if the dot P value is more than 0.7, that means it is very much reliable data. Here we can see in the COVID positive samples, the peptide for the particular protein is present with the dot P value of 0.9, which is a very reliable data, and thus you can actually see and actually validate your your hits that you get for your global data or different biomarkers are thus validated. Thank you.
All right, thank you, Amrita. So again, this was a bit complicated to really convey in a very short time. But idea is that let's say you have identified 5 proteins which looks very differentially expressed to you, and of those 5 protein, maybe 3 peptides each. So 15 peptide you want to analyze, and what you are seeing that only now you are looking at intensities of those 15 peptides across your control and treatment group. And if you can utilize that information, you can have a cumulative value of these, let's say 3 peptides of each protein, and now you can show what is the fold change happening at validation step. Is that matching the trend of your discovery set? And this can be a good way of you to move forward for the even absolute quantification eventually. So what we have tried to give you a glimpse of it. Proteomics can be done in different ways, different possibilities. Sample prep is the most important, and the data analysis, of course, the most important at the end. In between, there are different creative ways of separating the complex proteome. Given that we are talking about 20,000 plus human protein and maybe a lot of modifications and many other changes, so therefore we have challenge of separating these complex proteome and their modifications. And could be sometime gel, could be technologies like mass spectrometer, or even other technology like protein microarrays or biosensor like surface plasmon resonance, biointerferometry. Many of these can be equally involved and utilized to really understand the complexity of the proteome. So I hope you are getting excitement of studying these technologies, which will have good utility not only just for studying the clinical problems, but also for any biological problem for that matter. You can creatively ask some interesting question, and these instruments and these technologies can provide you the at least some clues and some glimpses of the potential possibilities, which you can now hypothesize and take forward for validation. So that's the a broad nutshell of the gel-based to the mass spectrometry-based proteomics workflows. If there are any specific query which we have in the forum, if somebody wants to ask something, TS can tell me. We can discuss that. Otherwise, we are already having the live weekly session with all of you. Also, we have the forum on your each courses when you are asking the question, and our TAs are trying to give you the further expression of that. Those are different ways of interaction. If any specific question for me, I'll be happy to talk to you now. Ankit, is there any specific query in the chat?
No, sir. No such
All right.
specific.
So anyway, I hope you are able to get a good feel of how these technology which we have been talking in the past can be done in the lab and the possible setup for those. This could be a good beginning for interactions, and we'll be happy to connect with you for another live session in the next month. All right, all the best everybody, and have a good evening. Bye.